All right, we'll get started. It is my great pleasure and honor to welcome you today here to the opening of Genetic Reflections. So my name is Anna Scope. I'm a professor in genetics. And so this is a collaboration by myself and also an artist, Angela Johnson. So I'm going to start this off. So we have a live web stream. So this is NSF funded. This is going to be disseminated everywhere. So we're going to talk about our collaboration, about who we are, and then after that, everyone's free to go out to look at the piece, and then we have an interactive piece that we'll tell you about at the end, that if you have kids too, you can also bring them. They'll be out there in a minute. So without further ado, so before I start, it's always nice to give a little background of why I do what I do. So I'm a scientist who also does collaborative art, but I need to tell you, because I'm a geneticist, I need to tell you about my genetics. So for most scientists, my background might be quite unusual, but I grew up in a family of artists. So here's a pedigree. I come from a family of artists. So my father was a sculptor and medical illustrator. He taught anatomy to medical students, also equally odd, but that to me growing up was very normal. And my mom was a ceramist, and they gave birth to 4 kids. I'm the oldest of 4. And if you know anything about genetics, your probably first thought there is that I may have inherited a recessive gene for science. But when in fact this is actually not true, is that what we inherited is a really amazingly sense of creativity, and creativity is really important in science. And once I got to meet scientists, I realized, wow, they're just like artists and super creative and awesome people. And so, but my 2 sisters do graphic design, and my brother's an industrial designer. If you don't know an industrial designer, it's sort of the person who designs the iPhone. That's an industrial designer. And so I grew up in this household, but I also was interested in art. So I have an art degree. I have a ceramics degree myself from an undergraduate. But I wanted to show you what my environment was. So to tell you why I do what I do. So this was my home growing up. So my father is sculpting the bust of his football coach at Syracuse. So my father played for the Syracuse Orangemen in the 50s, and he went to the Orange Bowl. And this is one of the most winningest coach who's in the Hall of Fame, Ben Schwartzwedel. So this is my home. Anyone who knows anything about science, this is a non-sterile environment. And I grew up in lots of dirt, lots of clay in my home to the demise of my mother. So this is my home, right? So you can imagine becoming a scientist was probably a really difficult trip for me, because this is what my life was like. My father painted in charcoal. This is a self-portrait of himself. And so I work on the process of cell division. So if you go from something like that you've just seen the art, and now you see something like this, your first thought is, this is amazingly beautiful too. And so this was what caught my eye as an undergraduate. I was like, wow, this is the most beautiful thing I've ever seen. It's like a Picasso. I wanted to figure out how that works. And so what my laboratory works on is this beautiful process. And the reason why we work on it is that when you have failures in this, it leads to tumor burst defects. It's important for how stem cells divide and differentiate. It's also important for nerves and brains in the cells. They have to divide asymmetrically. And so those are working on my lab. But inherently beautiful process. And as I began working on this throughout my scientific career, it was very clear to me that I wanted to share this. So we weren't sharing this enough. This is federally funded research. How could we share this more effectively? So as a graduate student, I started the C. elegans art show, and taking the images that we have in the laboratory and disseminating them more widely. So if you've been in the foyer in genetics, so when I came here as a faculty, I had a little more power and a little more gutso to do more things. So I started from that C. elegans art show doing big, large installations. And so this is over in the other foyer if you'd like to look at that. And from that became interactions between the art department, of which I'm also associated with now. And busloads of kids come through here and get excited about actually the beautiful work that's going on in our department. And from that came this tiny scientific art show that was a collaboration between Tandem Press and the UW-Madison scientists. And these are images from this. And these were on display at the Dane County Airport, and it's probably one of the most popular shows they ever had. It was up for a year. And we had a bowl of comments, and it was overflowing. People were amazed what stem cells look like, or how beautiful fruit flies that eat bananas are. it was an amazing thing, and it got quite a lot of press for campus, which was great for the research and discoveries that are here on this campus. A lot came from that, and I just want to share a few of those things. So I know Wei Hua is in the audience here. So one of the things that come out of science, so this is one of the most beautiful things I've ever seen. And so what does it look like to you? Monet's water lily. That's what's so amazing about some things you find in science that can't be published in a science journal actually are great for interacting with the public because you can engage them. What this actually is muscles and blood vessels in the eye, if I'm correct. Most beautiful thing in your eye is actually a Monet. And so this is really great for engaging the public with science. I think this won the grand competition. It's just an amazing image. And then this led to our Cool Science Image competition, which we've been doing through collaboration with the iResearch Institute. And I want to show you some of those images from past years that are quite remarkable. So here's the tongue, which I found amazingly beautiful there. Gold melting, your cerebellum, things that might not show up in journal articles, but are very interesting and engaging with the public. Here's our 2018 winners. More muted colors this year, but there's blood vessels in the brain again. Tungsten, unbelievably beautiful. And indium and cobalt make these unbelievable spirals, which I think are great. So what happened over time is as I was doing these things, art students were coming to my laboratory. So my first foray, prior to meeting Angela, I had a undergraduate performance artist, Chanel Matsunami-Gavreau, came to my lab and she said, hey, I heard you're free and open to have artists in the laboratory. I said, sure, come in. So she actually had a Guggenheim Fellowship. This is actually a big deal. If you're a non-scientist, you might know this is a big deal. She had a Guggenheim Fellowship in my lab to do this project called Differentiate, which is a very scientific term. She got interested in this organism I work on, C. elegans, and she looked at the skin and she actually made fabric based on the skin of the worm. And she did a whole performance piece about coming up and differentiating as you go through your life. And she was at the first art student to be at the undergraduate research poster session, and she did her whole performance there. So, and made it quite a splash. And so word got around that I was allowing more and more artists into the laboratory, and other people on campus have been doing this as well. And this led to my connection to Angela. Angela also came to me and said, hey, can I come spend time? So Angela did her MA work in collaboration with my laboratory, and we have 4 different scientists on our floor that do work on different model organisms. So her show is called Translation, which you could see on the 2nd floor. And this is one of my favorite pieces, which is outside my office. It's a zebrafish development. But it's very interactive and textural, and has a lot of movement to it that I really like about it, but also very scientific as well. So the connection between art and science is great. So I was applying for an NSF, this is National Science Foundation grant, and because of what I've known about Angela and what he did with her work, I decided to come up with an idea of how can we engage the public more broadly, and in connection with the UW Biotech Center, they were interested in filling this wall out here so students will be able to interact with the genome in some way. And so with the help of my brother, who's an industrial designer, helped me mock this idea up that we had to do this genetic reflections. And the idea is that you see the mirrors and cells inside of you and you reflect on this experience. And what happened the first time I submitted this, I actually didn't put budget in there, and then the second time around for submitting my grant, the NSF called me up and they said, if you put $50,000 in, we'll fund your science. And I said, well, that's an easy revision. and so I was quite shocked by this, so I revised it, and we put it in and we got the grant. And so not only funding my science, we're able to have $50,000 art piece, which is unbelievable, amazing. Not just the art, but also the work that we'll tell you about how we're going to disseminate the work more broadly. So this was sort of the initial concept, and I will let it over to Angela, who would tell you a little about how things transpired from there.
Maybe. Hello. Can everyone hear me okay?
Yeah.
Thank you for being here today. Again, my name is Angela Johnson, and Anna talked about her genes. I want to tell you a little bit about my genes. I have my dad's eyes. I mean that figuratively and metaphorically. They're dark brown, but also he had the ability to remodel every room in our house growing up, to my mom's sort of demise. But he would, on grid paper, draw what he was going to make, and he'd bring us all into a room excited. I'm going to tear down this wall, and I'm going to put this here, and do an arch, and do glass block. My mom was glazed over. My brother's like, can I go play Nintendo? And I was right there with my dad. I could see it. I could visualize it. I totally saw it in my mind's eyes. So again, I have my dad's genes. And I was right there by his side, helping him paint walls, grout, learn to lay ceramic tile. He said I cut his work down to double. He was probably right. Even when I was helping him build a deck when I was a senior in high school, I accidentally bonked him on the head with a 2x4. There was a fair amount of blood. But he still continued to teach me, and I feel that he was really my first collaborator. And on that note, I've always had a strong curiosity for visualizing, for creating, constructing objects that don't exist yet in the world, coming up with an idea and figuring out how to make it into a reality. And I've always had a really strong interest at the intersection between art and science. Anna had mentioned my MA exhibition was here up outside her hallway. My MFA exhibition, which is Masters of Fine Arts for those of you maybe not in the art world, was at Central Public Library and was called Inherent Legacy. So again, thinking in terms of family, genes, dynamics, I turned my exhibition into a cabinet of curiosity, or a natural history museum installation, so to speak. I took old family images, and I took new images as well. I'm trained as a photographer, and created these light boxes that are multi-layered. So working on alternative types of materials. So I took old family wedding images. I took close-up installations and close-up shots of family artifacts. One of my great-grandmothers created an embroidered piece with months and flowers of the year, and put those in cherry wood boxes that are meant to kind of represent windows that you're looking in or out of. Behind that was frosted plexiglass with color-changing lights. So there's this push and kind of pull of the imagery where the back image looks like it's moving forward and back, and this really interesting passage of time. There were also colors associated with that too, so you could possibly think of maybe blue might be a sadder period in life, green might be more prosperous, so sort of moving through a journey, so to speak. This is another piece that I did called Grandmother's Stories, showing that I really enjoy layering work. My grandmother had kept, she'd written stories over the years and made binders for each person in the family, and stories of when my grandparents owned a hotel in the Dells in the 1950s, stories of my great-grandfather being the blacksmith in Montello. And I created this book using alcohol transfers, so I scanned family images and put them on canson paper, and then in my handwriting, but her words, put the stories behind. So again, it's interactive. You look at the images on, excuse me, the outside, look to the stories on the inside. You can move around the podium and see a whole nother different set of stories. So again, showing you layering kind of multiple images, different ways of looking at and engaging work. Printing on mirrors was something I also did for the MFA exhibition. I had an old 46-inch dresser mirror that was printed on. It was not an easy thing to install at the library. But the exhibition was on the second floor of Central Public Library, which houses local history. And this was an image of my great-grandmother Pearl and my grandma Claire, taken in 1918 on the east side of Madison in their house near Tenney Park, to give you kind of a contextual place. So the black parts are inked and filled in, but the the silver or white parts you can see yourself in. So again, the second floor is where the local history is stored. So you can see yourself, you can look to the past, both at the image and at the stacks of the Hall of Record behind you. As I mentioned, I used alternative photographic processes, printing on mirrors and layering, and Anna and I had an interesting time talking about the possibilities for this exhibition, which we are calling a year of experimenting. The hallway, it may have been mentioned already, out here, was very specifically determined for this project because the outreach lab that brings hundreds of K-12 students to the Biotech Center to learn about DNA and experiment and extract wheat and other lab experiments that they do, walk directly through this hallway. And Dr. Tom Zinnan takes students out and goes to the double helix metal water fountain sculpture and talks about DNA being a recipe with repeating codes. And so we thought, what a great space and opportunity to add to that story, which I'll elaborate more on in just a little bit. So that was why that hallway there. I'm going to talk a little bit through, we spent the fall doing test samples, figuring out what will work, what won't work. I had tried some of those alcohol transfer methods. I've done some gel medium methods. Turns out those don't work well in mirrors at all. But much like scientists who work in a lab on experiments, sometimes you really need to try and find out what doesn't work to get to what does work. I'm also going to talk about lab meetings and how we I immersed myself in Anna's lab and would talk about my progress and get feedback as we went. And then also how I was able to work with Doug Powell, who's here in the audience, to work in the makerspace for what ended up being our final piece. Again, a year of experimenting. There was so much math when we finally got to what we wanted to do. If you haven't been into the hallway yet, you will see. This was such a collaborative project. Initially, we were going to, from the mock-up you saw that Anna's brother had created, we were going to do mirrors directly mounted to the wall. We were going to work with a printmaker. I was going to print them myself. We were going to use biological images, and that was our plan. Some of the methods I used didn't work as good as I'd hoped. We worked with a local printer. They were interesting, but they still felt stagnant. Images on a wall, it didn't feel that interactive. So we moved away from circles that were referencing a petri dish to Anna had worked with Guy Plunkett III, who had worked with DNA sequences for 10 different model organisms, and we thought that would give a nice representation, still honoring the words genetic reflections, where people could see their reflection, but it wouldn't be in a mirror that had been printed on. So once we got to that point, my husband and I came here one snowy Sunday evening. We pulled out the tape measure. We had 53 feet we were working with. We were going to move it down into 40 feet of installation space. And the conversation started out, I want each glass panel to be about this big. Well, 2 feet, yes, 2 feet. And then determining how much space do we want in between. Then we went to how long bay for dinner, and on napkins and in a notebook, we were writing down what was going to go where, and the waitress was really interested, like, what are you guys working on? But from there, my husband Justin worked with me to create this map, again, working really collaboratively. You can't just install your own thing in the hallway of a university building. You need to work with an awful lot of folks. So this really required accurate measurements to be able to give to the carpenters who were cutting their Baltic birch and to the glazers installing the glass. We wanted things 57 on center, that's a gallery term, for the glass and the mirrors. Since it's a hallway that so many people go through, even little ones who are in kindergarten and first grade, we still wanted them to be able to see themselves in the reflection. up to tall people. But if maybe you're 6'5", you have to scoot down. Sorry, if you are. But this was really important to have completely accurate. The folks from physical plant had it taped to the wall opposite, and literally were working directly off of this. So again, in collaboration to make sure everyone had the recipe and steps necessary to make sure it accurately went up. As I mentioned earlier, we had weekly lab meetings to make sure things were on track, to run ideas by, to see how my progress was coming. And I want to give huge thanks to all of the physical plant staff. Again, there were probably 10 different folks, from the electricians to the painters to the glazers to the woodworking installers. We also created templates for Martin glass. We wanted a specific curvature to the glass. We also wanted a specific curvature to the wood. One, it's a hallway that a lot of people go through, and even though it's art and you shouldn't touch, we know that, kids walking through, what's the first thing you do? You're going to touch. So we wanted to make sure that everything was streamlined, but also thinking about the 10 different model organisms, and any model organism, including humans ourselves, we are also streamlined and organic and have soft curves. So that was intentional as well. After we decided we wanted to do the DNA sequences, what was the best way to show that and visualize that? And the idea that we came up with was laser etching. And just about a block and a half away, the UW Makerspace and the engineering library has a laser cutter, which is amazing. And I was really excited to get started. And when I reached out to them, they said, you have to be an engineering student or you have to be affiliated with the engineering department. I said, well, years ago I worked with the MRSAC, and that's great, but you need to be affiliated or partner with someone. So that's when the MRSAC was wonderful enough to let me work with Doug Powell, who's in the audience. He is right in the middle. He is an engineering undergrad and also getting a certificate in art. So it was a great partnership. We, for a good 2-3 months, tested on the laser cutter. Thickness of lines, different fonts. We ended up using Arial font because that's universally readable. Different densities, opacities, in terms of the organisms, etching them to see how they rasterized and how those would turn out. So much time, again, as a scientist in a lab, experimenting to see what we can get for the best possible product. After we determined what worked best, we had all of the glass that had been ordered. We put it down in the laser bed to get started. And Matt Norman, who is all the way in the back, he's our videographer who's been working with us and is wonderful, was right there that first day, kind of over our shoulder as we were lifting up the first panel of glass, which was equally exciting and slightly terrifying too. That like, oh, how does it look? How does it look? And we're being video recorded, smile. But it was really, really amazing. But it was also just a really interesting experience to go through after so much experimentation to see what the final product would be. And there's me staring very intently. And this is a 2-minute and 14-second video that Matt put together that will make it look like the process went just like that. So thank you again to Matt. He made that look so easy. The process went together seamlessly. So that kind of gives you an idea of the process of the last year working on this. And if you haven't already seen it in the hallway, here is the piece up just outside the hallway to here. And with that, I'm going to turn it over to Anna for just a moment.
So before you go out there, I'm going to tell you a little bit about the science of what you're looking at. So the sequences we chose, and in collaboration with Guy Plunkett, we decided to choose sequences that are used to compare organisms in the world. And so those are called ribosomal RNA. And they're actually, how they fold on each other are actually quite beautiful, but we just used the DNA sequences here. And so the red arrows are showing where this RNA actually is. So the ribosome is actually a cellular machine that allows you to make proteins in the cells. So all organisms use this machine to do this. And this sequence is common to all organisms. And so it's actually quite similar. So when you go look at the sequences, you'll see, oh, a lot of the sequences look quite similar, but there's actually fine differences between them. And so that's sort of the idea here, is that you could see yourself and how similar you are, but also other organisms have the same thing. So it's sort of the concept here. And so what we're going to do is be able to show how, and to young students and also the public, how these all are related. So you take these sequences and you can build these things called phylogenetic trees to find the relationships between the organisms. And the order in which we chose the 10 model organisms up there, a lot of them are used on this campus, and the order of the art piece is based from the top. So we'll start from Baker's yeast, Saccharomyces cerevisiae, down to E. coli, which is a prokaryotic, which is actually a bacteria. So when you walk down the hallway from left to right, you're going from yeast to bacteria. So that's how the relationships of the art piece are built. So what's actually going to happen next? So the next steps is we have to create a traveling piece. So part of the NSF Broader Impacts is actually to have something that goes wider. And so this traveling piece, since this piece is very big and is actually permanent, has to be bolted to the wall and probably more difficult to ship than anything else, we decided we're going to build a piece that's easier to ship and travel in a lot of places. And so we already have several universities already lined up, Lawrence University and Sloan Kettering in New York, other places. And so if you know of anyone that's interested, we're also interested in having this travel quite broadly. I think we're also out for a couple years already. And then we will work with Tom Zinnin to actually build an interactive website so that students who come here will use their phone and take a QR code and be able to get the sequences and build trees. And so we're going to work with Tom to do that as far as disseminating this widely. And maybe students across the state of Wisconsin will come here to see this and take it back to the classroom. And so the goal here is actually to be a beautiful piece about genomics and evolution and genetics and how we're all related, but also be able to teach students and impact not only the local community, but across the state and the world for that matter. So without further ado, we have so many people, we can't do this alone. We'd like to thank a lot of people on the list, particularly people in the Laboratory of Genetics. A lot of them are here. MRSEC, the installers, the Biotech Center. So we didn't realize how much it would cost to bolt things to a wall. At the last minute, they pulled through. So in order to take out the wall, it was going to be $17,000, which we didn't budget for. So Biotech Center came up to plate, and we found a new way to mount this with this wood, which is amazing. So I thank Chuck a lot and Tom for a lot of help, and especially the art department and the campus community and family. Is that right? Anything else you want to say? Here, it's good. So we'd like to invite you out to the piece. We have food and drink, and we'd like to welcome yourself and also go into 1340, which is just at the end of the piece, and you can have a little takeaway silkscreen bookmark of your favorite model organism that's up there. So, people from the Art and Craftsman Supply are there to help do that, so I encourage you to go down there and do that, and thank you again for coming. And we're welcome to take any questions if you have them. Thank you. Any questions? Yes?
Where was the installation that you showed, the intermediate installation that was in a museum?
Oh, that's actually a mock-up picture we made for the grant. It's not a real thing.
Was it Photoshopped?
It's done. My brother's that good. That's what he does for a living. That's in CAD, I guess, a drawing. He mocks stuff up virtually. Like it looks pretty good, actually. So that's what got me the grant. So that's
calling the inspector.
Yes. Thank you for that. Any other questions? If not, help yourself to the food and drink, and thank you for coming.